Okay, so here is my attempt to demonstrate where the AY-AT recombinant came from and why it also produced an AW chromosome as part of that event. So first I'm going to talk about where the different alleles came from of A, and I'm going to represent the chromosome on which the A gene is found by these pieces of paper. So this is chromosome 24. And here is the A gene with two parts of the gene that I'm going to talk about. And actually this is, of course, is drawn nowhere near close to scale. So a better thing than a piece of paper would be a string of beads. And if so, chromosome 24 would be 50 million beads long. And the A gene would be about 39,000 beads within that 50 million set of beads. Okay, so I'm showing the A gene here as being a big part of chromosome 24, but of course it isn't. It's 0.07% of it. And if this was about an inch, then actually this piece of paper would be 108 feet, so way bigger than it is. So the four alleles were derived from a chromosome containing AW, it's thought. One of the first, the AY allele, came from changing a couple of these beads, so two of these beads to a different one, so that instead of in the protein instead of the amino acids alanine and arginine, which are found in the AW version of the protein, instead you find a serine and a histidine, and that's the AY allele. The AT allele came about when there was an AW chromosome, and an additional sign sequence. This is an inserted piece of DNA here that's found in the AW allele. An additional one was inserted into that chromosome, and this makes the AT allele of the chromosome. And a chromosome that had AT allele on it, there was an additional change that changed one of the beads that are found in the A gene to a different one to cause an amino acid sequence change, and then this is the recessive recessive black version of A. So there are four known versions out there. The original AW, which gives wolf sable, the AT version that has the additional sign sequence, and that gives the black and tan, recessive black, and then the sable. So the question is, how do we get a recombination that has an AY and AT on the same chromosome? And that came from the fact that during the process of sperm and eggs, we always have recombination events that take place. So everybody knows that you have two chromosomes from two of each chromosome, one that came from your father and one from your mother. And during the process of making sperm and eggs, these get separated into different sperm or eggs, so that you're passing on the chromosome you got from your father or from your mother. However, a part of that process called meiosis is recombination. And recombination occurs, now this is oversimplifying it, but this is basically what the end process is that the two chromosomes get cut at the same place, the same equivalent place, and then they are reattached, but instead of the red chromosome getting attached back to the red chromosome, instead it gets attached to the equivalent place in the blue chromosome, and vice versa, to generate two recombinant chromosomes. Now, this is actually a necessary part of the process. 1 to 3 recombination events take place on every chromosome, and if that doesn't happen, meiosis actually stops, you don't make sperm and egg, and the cell actually goes on to kill itself. So, in addition, so like I said, there's, will be 1 to 3 for every one. I'll show 2 on this particular one that take place, and where it takes place is pretty much random, but you always generate 2 recombinant chromosomes, and then this would be put in one sperm or egg and this would be put in the other sperm or egg. Okay, so what must have happened to make the AY-AT allele is that there was a dog that had AY on one of its chromosomes and AT on its other. Now normally, remember, this is like an inch, right? And then there's 108 feet. There'd be 1 to 3 recombination events taking place in this pair, but most of the time the cuts would take place outside of the A gene. However, if a cut took place in the middle of the A gene, in between where the extra sign is and where this amino acid change is, and then these are then attached in this way. Now we've made a new chromosome that has the AT extra insertion, but this amino acid sequence that corresponds to the AY mutation. Okay, so this would be the AT-AY recombinant. Now notice that the other chromosome looks the same as the AW chromosome, just because it has a single sign, and then it has this particular amino acid sequence there as well. Now once that recombination takes place, these are just passed on to offspring like the other alleles on the other chromosomes. So the dog that gets this particular recombinant AY-AT chromosome, so let's just say that the other parent contributed an AY, well, now when this dog goes on to have offspring, 50% of the time it's going to pass on the AY-AT, and 50% of the time it's going to pass on the other AY. So although the recombination between these two parts of the gene is very uncommon, very, very rare, only 1 sperm out of 1,000,000, or I don't know the exact number, even that's very, very rare. Once it's made, it's going to be passed on just like a normal allele. So hopefully that's helpful. And if you have any questions, you can put them in the comments.